welcome everyone to this first long workshop given by Stefano Mangiola. I hope I pronounced it right. So he is part from the WeHi Institute, located in Australia, and I'm going to give today a workshop about the tidytranscriptomics, so how to use the tidy paradigm for doing transcriptomic analysis in R. So the floor is yours. I'm looking forward to this interesting workshop.
Right, thanks, Christoph, for the introduction. Welcome, everybody. I hope you will enjoy this workshop. In this workshop, we will talk about both single cell and bulk transcriptomic analysis done in a tidy manner. So before we start, I will just give you a few resources. One is the actual website of this workshop that is available online for you to, there is the full render of the workshop. And so will help you possibly now if you want to see the render version or for future use. And I asked Christoph to send you in chat the link of the website.
Yeah, I'll do that immediately.
So also there is, I will ask Christoph to send the GitHub repository where there is this workshop, all the many of the tools we will talk about today, and as well other 5-6 workshops we have done on the topic. So we vary content every time, so there is plenty of resources for you to choose from for future learning. Okay, beside that, before I start, I would like to ask 3 questions to the audience. So I will launch a poll and you can reply yes or no. So the first question is if you had previous experience with bulk transcriptomic analysis. I want to know how many of you have done already bulk transcriptomic analysis and are familiar with that. Okay, so right, more than 70% is good. I will stop it now. The second question is about single cell. So how many of you had previous experience with single cell transcriptomic analysis? So I know how much background do I need to give before I start with the actual analysis. Okay, this is 50%. So half of you had already experience with that, 60% close. All right, thanks for that. And the third question is how many of you did do analysis with tidyverse language before? So are you familiar with tidyverse packages such as dplyr, tidyr, ggplot, so on and so forth? All right, great. So 80% of you are familiar with tidyverse. This will help a lot, of course. All right, thanks for that. If you have any question anytime, you can ask it. It will help also me staying awake because here is night. So help me make this workshop quite interactive, and I welcome any question anytime. All right.
And sorry, I didn't mention it, but you can ask questions either in the question and answer tab on the right, or you can also raise hands and I can invite you to the stage if you want to have a more elaborate question that you don't want to type.
Great. So in the background tab of the website, at the end, there is actually the link for the cloud to access the RStudio session. And here we have a screenshot because we have quite few tidytranscriptomic workshops in the kind of bioconductor system. You need to choose a specific one, which is called tidytranscriptomics for bulk and single cell analysis. And it will be one of the ones with not so many launches, okay, since it's a new one. So you click on launch and you will be sent to a waiting page where the virtual machine is being set it up, and then you will be able to access the actual RStudio session. If you have any question about that, please let me know now. the RStudio session you will be able to access is the same as the one you are seeing on my screen. While you are connecting to RStudio, we'll just give a brief summary of the content of the workshop. So today the workshop will be divided in 3 parts. The first part will be bulk RNA sequencing analysis powered by TidyBulk. The second part will be just a 2-3 minute long and will be just an announcement of a new tool we have produced that is called Tidy Summarize Experiment. And the third part will be about single cell analysis and will be powered by the bioconductor workflow and a package that we have developed called Tidy Single Cell Experiment. Okay, I believe some of you might already have access to RStudio. So to code along, so to execute the code I'm executing, you can access the VicNets folder and the tidytranscriptomics.rmd file. Okay, so I will launch another poll. I would like to know how many of you are already inside the RStudio session. All right, can I ask a question? How many of you have now accessed RStudio session?
So I see someone says it's taking quite a while to launch.
Yeah, we are quite a lot today. So I imagine 70 virtual machines are quite a big job to start. Hopefully, none of them will fail. I think that the actual computer, the machines they are running on are quite powerful, so they should handle all of them. Yeah, one thing I can do, I can start with the actual material, and people will be able. I'm happy to stop anytime if people feels I'm going too fast, and people will be able to once inside to run the code, until you know the point we are at.
Yeah, exactly. I see also Asa is asking, can I follow it by downloading the repository and work locally? Is that something?
You can. It will take time because there are dependencies, but on the GitHub page of the repository, I will show you. Sorry, I will show you here. If you go to the okay, we could even share the link of the repository, which is in my GitHub page, BioC Europe 2020. There are all the instructions that you can use to install the workshop locally using install GitHub from RStudio, from R, or even with Docker. So yes, it can be run locally, definitely, but at the moment, I think it also might take some time. So I will try to share where, I guess, in the... Christoph, I will send it to you.
Okay.
Okay, this is the actual link of the GitHub repository of the workshop. Okay.
Okay.
Let's start. Otherwise, we have to go through a lot of content. So I don't. I know it will be not actually easy to stop for a long time to ponder on the code, but I will give a slow pace. And this workshop has plenty of information, so you will be able to come back to this workshop and the actual render version in the website and really absorb all of that. But I welcome any question anytime. Okay, so let's start with the bulk analysis first. I assume again most of you know how to do a transcriptomic analysis. This is not really a workshop on how to perform a transcriptomic analysis, but how to perform them in a tidy-friendly way. So first of all, we load all the packages. For this bulk part, we will use the airway dataset. In the meanwhile, we load also the high statics for the ggplot. That's not mandatory, but we will get consistent styling then. As I said, we are basing our analysis on our airway dataset that includes 8 samples, 4 cell lines, and 2 treatment conditions, and includes the actual data is from human airway tissue. So if we visualize this airway dataset, you can see that is a summarized experiment object, which is a data container for bulk transcriptomic data from bioconductor. And we can see here that is giving us some summary information. Okay. Using tidybulk, we can represent this data into a table form. Table is a advanced form of a data set, of a data frame from R. And we can do it applying the function tidybulk from the tidybulk package. And we can see here that the data we have is features, which are ensemble IDs, samples, row counts, and a few information about the samples and other information as well. Okay. And so tidybulk not only allow us to represent transcriptomic data as a table, effectively this is a table just with few attributes added to it, but also provide us with a workflow that is respect the tidy paradigm. So same input, same output, and is quite modular. And as you will see, it uses a quite clear self-explanatory grammar, so not acronym and no specific jargon, okay? Because the goal is really to make it easy for even users that are starting to understanding better what analysis they are doing. Okay, so as an example, we can use already some of the tidyverse tool to polish our dataset in a pretty intuitive manner. For example, let's say we want to remove this prefix from the sample column, and then we can simply use mutate on the sample column, removing this string. And we can also create a new column called symbol, which it will have the gene symbols, and we can extract them directly using this function, map IDs from annotation DBI, from the ensembleized IDs.
And Stefano, Niels is asking, is tidybulk in any way related to BioBroom?
No, it's not related to BioBroom. They have kind of similar goal in mind, but they're quite different, totally independent. Okay. Yeah. And so a quite neat thing of tidyverse, and as most of you might know, and also of this way of doing analysis, that we can pipe different procedures one after the other. We don't need to create so many temporary variables if we don't want to. So the point is we create variable when we are using one object more than once and where is convenient for us. But we can perform complex operation without creating any temporary variable. And you can see here that we are popping all these commands together. And what we get out of is our clean sample name and our symbol column at the end. Okay. All right. So this is just an example workflow, but I will go through few key steps of a standard bulk analysis workflow. We start with data filtering. Usually we want to filter lowly transcribed genes, both because they don't carry so much information and because statistical inference algorithms are not so robust and designed to analyze lowly abundant transcript.
So for people that are still trying to connect to the orchestra, so it takes some time to launch. Once you launch, it will ask you an email or login and a password. And yes, if correct, both are RStudio.
Correct.
Lowercase letters and in one word. Okay, yes. That's for the ones that are still, because I get some questions about it.
Yeah, that's great. And again, once you get in, go to the vignettes folder and tidytranscriptomic.rmd file. Okay, so to filter data is quite straightforward with tidybulk. We just call keep abundance, and we can provide a factor of interest, in our case treatment, that will help inform the filtering also based on our grouping. Using an HR filter by expression function on the back end. And we can see here that we have roughly decreased our data set size of 5-folds. For example, if we want to know how many transcript we are left with, we can simply use some tidyverse syntax. We have 13,000 transcripts left. Okay, so another important step of bulk analysis is scaling the data. Is also called with the jargon normalization. And we need this because we are carrying the technical variability from differential sequencing depth of different samples. And so we want to make all the counts consistent for visualization purposes. And also when we do some hypothesis tests, this scaling will be done also for this step implicitly in the statistical model. But in our case, for directly scaling the data for visualization, we can call scale abundance. And this will use different possible methods, but by default is using TMM from the HR package. And we can see here we get out the same table, but this time with few columns, few additional columns, which is TMM factor, the multiplier, so the scaling factor, and a new column called count scaled, which will include our scale counts. And one first thing we might want to know if is the scaling procedure has worked. And so we might want to plot the densities of the transcript abundance before and after scaling. And so we can do this with a custom ggplot quite easily using the geom density function. And a quite neat that you will see, a quite neat thing that you will see a few different times through the workshop is that it's quite easy to produce faceted and quite complex and informative plot with this kind of way to do things. And so, for example, reshaping the data on the row counts and scale counts, we can then use this new column to build a faceted plot. Okay, so this pivot longer will transform two columns into one, and the actual counts will go into the abundance column, and the before and after normalization will go into the source column, and we facet based on source. As you can see, we are also coloring by sample, and we can see that the scaling procedure has worked, at least visually, as all the density, they look consistent to each other.
I have a question here from Felix, who is asking if tidybulk is the central melt function of the data.
Is the central melt? I don't understand the question.
I think it has to do with, there is a melt function. I don't remember in which package it is. Well, it's able to put everything in a long format.
Okay, so, well, better I clarify one thing. Many of the data manipulation functions we are using here are straight from dplyr, IDR, ggplot, and so on. Okay, so this melt function that he's talking about is probably referring to this pivot longer. They do similar things. In this case, they do the same thing. So they reshape the data from wide to long.
Okay. Exactly, and Aydin is saying melt is actually from the reshape 2 package.
Yeah, exactly. Yeah. And yeah, so the function actually from tidybulk are just the few I mentioned. Is, for example, keep abundant, scale abundant, and so on. So I will try to be more specific which functions are from tidybulk, and the rest will be actually from tidyverse ecosystem. Okay, so again, because we can build quite
sorry, there's still a follow-up question now. Is there, so Aidin is asking, there is also wide to long functions in the multi-assay experiment package. So I guess that's for those that want to have different assays and maybe different omics or whatever. Okay, right. sorry for the interruption.
No, interrupt when you wish. So again, we don't need to create any temporary variable to visualize the data and manipulate the data. Here we can do whatever we have done so far. So tidify the data, filtering, scaling, and reshaping and plotting all in one go. Okay, and by comparison, this is if we want to use just base R, and I'm sure there are ad hoc functions to do this, but we can see here that the code is quite a bit more complex, and we also have to create quite a few temporary variables. Okay, so another important step of bulk analysis is exploratory analysis. We want to understand the technical and biological variability of our data. And so one important step of this exploration is dimensionality reduction, because we cannot visualize high dimensional space. We reduce the 13,000 dimension, one for each genes, in just couple that we can visualize. And one algorithm to do it is PCA, and there is a tidybulk function conveniently called reduce dimension that can do this. There are other dimensionality reduction methods such as t-SNE and UMAP we can choose from. And in this case, by default, we calculate the first two dimensions, and we can see here that the printout tells us the fraction of variance of this dimension. And the data we are returned with is, as usual, the same table, but this time with additional column, PC1 and PC2. Okay. Because this data is, some of the columns are redundant because we are both representing samples and genes in a long format. If we want to visualize and plot just sample, for example, sample-wise information, we can use the pivot sample function from tidybulk to extract just the sample-wise information. So in this case, we get a dataset with 8 rows, one for each sample. And this includes, of course, also the PCA we have calculated so far. And for example, we might want to plot this PCA, and we can use easily ggplot if we want to customize the plot a bit more. Again, there are plenty of ad hoc functions to do this. And so in this case, we call pivot sample, and we produce a scatter plot with PC1 and PC2. And we have colored by treatment and shaped by cell type. Okay, treatment is our factor of interest, cell type is our confounder in this study. And as you can see, as we would hope, the PC1 described pretty well the treatment condition, and the PC2 described mostly the cell source. In this case, the square cell here is quite different from the other three. Okay, so we can realize what variance we get about the biology and about the other part of our experiment we might not be interested in. Okay, so another example of quite useful exploratory analysis is hierarchical clustering and heat maps. Again, one way to do it in a tidy-friendly version is that we can use the function from tidybulk called keep variable. This will filter just variable genes, and we choose to filter the top 500. And the heat map package we will use is called tidy heat map, is on CRAN. And this will allow to input any table and output an annotated heat map, of which we can add annotations simply in a modular fashion, simply writing down the columns we are interested in. Okay, so as you can see, the argument here are sample feature and the value are count scaled. And we want to transform with log function. And we add both the treatment and the cell sources annotation. And as you can see here, we get kind of the same picture where samples are clustered together according to treatment, except for this yellow cell that again is quite different from the rest. And if we wanted, we could get deeper into which genes are preferentially transcribed in one cluster versus the other for deeper interpretation. But this is just to show you an example of how to produce a annotated heat map in a tidy fashion. And so as you can see here, we are integrating tidyBulk and tidyverse function quite effortlessly. And this trend will continue for the rest of the workshop. By comparison, another way to do it with base R is this. Again, this is just an example, but as you can see here, a lot of temporary variable are created, and there is a bit of higher coding burden here. Okay. So after data exploration, one common thing we might do in transcriptomic study is doing some hypothesis testing about whether there are genes between the two classes of interest that are of which transcript are differentially abundant. Okay, so
Stefano,
yeah.
Sorry, Eileen is again asks a question. She says, "Nice color scheme. Is it a good colorblind scheme?" So I think, well, yeah, for the column, yeah. Exactly.
Yes, okay. It's good question. Well, I think the actual heat map here is quite must be colorblind friendly, as is kind of a light and dark scheme, but is not from one of the packages that provide those kind of palettes. But is a nice suggestion. As I mean, I developed this package, so I will think about adding colorblind palettes as well. Thanks for the question.
Yeah, I might just a small reminder. So you can ask, of course, questions in the question tab, but don't hesitate raising your hand so people you can immediately ask the question live. Okay, yeah. Thanks.
And talking about colors, actually, you are free to change. These are the default color, but you are free to change color for any of those layers. So heat map layer and all the annotations as well. You can provide palettes that the colors will be picked from. Okay, so for differential expression analysis, there are many methods out there that you might know. DSIC2, EdgeR that has many kind of analysis streams. LimaVum, same, has few different analysis streams. And their syntax and the jargon they use is quite different. And so we end up, some of us, to choose a preferred method and to use that, even because the burden of testing one method after the other is quite high. And we expect the result to be kind of compatible. Well, tidyBulk offers a quite modular and convenient way to do this, abstract all these analysis, and we can call tidy differential test differential abundant function. And we can here specify our linear model through a formula that you would generally use to build your design matrix. We can specify methods. There are many available. Here we are just running 4 of them, and we can run a prefix as the prefix are really helpful to reshape the data and really build a visualization in an easy way. Here we are using, yeah.
Sorry, yeah, there is Francesc that is asking, is there a fast way to add the explained variance by each principal component in the PCA?
Yes, I get this question. Unfortunately, this is, we get the printout because it's not that trivial to incorporate that information in the tidy format we are using. and the PCA variance is neither relative to sample or transcript. So the answer, unfortunately, is not. But you can easily do it. Of course, it's not automatic, but with the ggplot, customizing the labels. Okay, so we perform, in this case, comparative analysis of 4 different methods, piping one analysis after the other, as the statistics will be added to the original table. Okay, so the input and output remain consistent through the pipe. And then I should mention that for hypothesis testing, PCA, and many of the procedures, the actual raw results will be stored in the object, in the table, in form of attributes. And when you do the procedures, actually, you will receive a message from Tidybulk telling you how to extract that information. So what is the name of the attribute that you can use if you want to do deeper analysis on those procedures. So as you can see, a few different columns have been added to our table. We have the statistics from our methods, as you can see here. And now one neat thing we can do, again, is to incorporate some visualization from the Tidyverse ecosystem. Here we have GG pairs. And we can select the p-value that have been estimated from the 4 methods, plot them one versus the other, and appreciate which method they provide similar results and which not. So the HR streams, analysis streams, which is quasi-likelihood and likelihood ratio, they are the most similar to each other. And we can see here that, for example, LIMA-VUM and DSIC2 are the most distinct. And then I don't show you in this workshop, we don't have much time, but you could easily select the transcript. Each point here is a transcript for which the methods do not agree, and pipe into again, visualization tool to understand what's the distribution of the abundance of those transcripts, to understand why methods treat them differently.
And Laurent is asking, is independent filtering reapplied when using DSEC2?
I believe the answer is yes. I should make really sure, but I'm pretty sure yes. So, for example, normalization and also scaling, now that I remember, in case of DSIC2, are performed internally when we perform this procedure. Okay, so as before, if we actively scale the data or adjust the data, this will not be taken into account when do hypothesis testing. Everything we need to know will be included in the formula. But again, the answer I'm pretty sure is yes in the case of this. But feel free to open some issues to ask questions also in the GitHub repository for allowing me to really reply in a certain way. Okay, so let's suppose we chose our methods and we go with that. In this case, it's quasi likelihood from. Actually, sorry. Here, this code chunk is show you how to produce some summary statistics. In this case, we are just counting the number of significantly differentially abundant transcripts. Okay, let's suppose we want to go with our methods. By default, this uses quasi-likelihood from HR. As you can see here, what I was telling you before, you can access the fit object directly that is present inside the attributes of the table. As comparison, this is what we have to use with base R for the same procedure using HR. Okay, so once we have done our differential expression analysis, there are many visualization tools we can use to appreciate the statistics that have been produced. One of them is volcanoplots. Again, many functions that ad hoc produce volcanoplots, but here I show you the case that we might want to customize the plot for publication-ready visualizations or data integration, so on and so forth. So usually, when we stay in the kind of cookie cutter path, is all good. But once we want to customize things, it's a bit more difficult. We have to extract information and maybe use ggplot. So, for example, using ggplot is quite easy here for a base visualization. We can see here that we are comparing log-fold change with p-value. A quite neat thing of ggplot that has a quite powerful scaling functionality. And so again, instead of transforming data, for example, calculating the minus log p-value, we can use the raw, the real data, the real p-value, just applying the correct scale. So communicating to the user more transparent information. Okay, in case we want to customize this volcano plot for maybe make it a bit more informative, we can select some top genes here that we want to visualize the text from. Here we are selecting transcript-wise information that will include, of course, our statistics, and we just select the top differentially expressed genes. And as before, we use ggplot, but in this case, we just select which genes we want to plot the text from, produce a scatter plot, and add custom aesthetics. Okay, again, this is just an example, but as you can see here, that we quickly went to a quite pleasing looking plot where we customize size, transparency, color, text, so on and so forth. And because there are so many packages linked with ggplot, really the possibilities are almost endless. Okay, so before to go to the last part of this bulk workshop, bulk session, I will mention a function that many appreciate. Because tidybulk tracks every method that is used through the workflow, we can produce automatic bibliography of our workflow, and we can import this text into our reference engines. And this is both appreciated by user because it's a time saver, but also by developers, as some of the methods we used are hardly cited, for example, for visualization or data manipulation. So we make sure also developers of second layer methods that we use are credited. Okay, so we have some few minutes. The last part I will show you is cell type composition analysis. Sometimes, for example, if we do cancer research, we might want to know what is the composition of the microenvironment. So what cell types are present in our data, in our samples. And so for this, there are different algorithms. Some of you might be more familiar with CyberSort, for example, which takes into bulk RNA sequencing data and outputs an estimate of the cell type proportion in there. For this part, we will use a subset of the BCA of the breast cancer dataset from TCGA, the Cancer Genome Atlas. As before, we produce a tidybulk table. We can see here that we have patient column, transcript column, counts, and some survival information. To perform the convolution is quite easy with tidybulk. We just called the convolve cellularity. Again, there are different methods available and different references, transcript references that we can use with the methods. By default, we are using CyberSort with the LM22 transcriptomic signatures, which includes 22 immune cell types. We can see here, execute this. The action get I don't have time to go too much into it, but every function in tidybulk have an action argument, and in this case we choose get we just get out the patient-wise information. So we forget about the transcript. And if it's a differential expression test, we choose action get, we just get out the transcript-wise information. So we don't have to do pivot sample, pivot transcript after the fact. Anyway, so we get out our data set with 22 rows, one for each patient. And we can see here that we have columns for each cell type with a convenient prefix. As mentioned before, we can reshape the data for visualization purposes and analysis purposes. For example, here again we use pivot longer, same as melt function has been mentioned before. We select the columns with the cybersort prefix and create two columns called cell type and proportion. Okay. Again, I didn't mention this function is from tidyr. So the function I'm using are either from tidyr, dplyr, or ggplot. Okay, so quite simply, we can create a really informative visualization. We can create bar plots, so on and so forth. In this case, I chose to create a bar. Sorry, I was mentioning we can create box plots and so on. In this case, I chose to create a bar plot. Where on the x-axis we have our patient, on the y-axis proportion, we color by cell type. And we can see here that this plot is quite informative as we can appreciate what might be the most abundant cell type. In this case is macrophages M0. And what are the samples that are a bit different from the rest? For example, these two, they have different tissue composition. Okay, so one really good thing about this way to do analysis that all the procedures are really modular. So we have some module that we can use alternative with different functions. For example, before we have used reduced dimensions to calculate the principal component based on transcript abundance. Nothing blocks us to reduce dimensions, so to calculate a reduced dimensionality based on tissue composition. Okay, here we are just filtering the cell type patient pairs that have all zeros for their proportion. This kind of is a fluke of cybersort. PCA don't work on things that have zero variance, so this is kind of trivial, but the key part is reduced dimension. We are choosing patient as our element, cell type as our feature, and proportion as our value. And in this case, we can appreciate which sample are similar to which, not based on straight transcript abundance, but based on their tissue composition. As you can see here, we have two samples a bit different from the rest, as we might have seen before. So here are some key points. I will not go through them. You can read, but basically is a summary of what I mentioned above. We leave 5 minutes for questions, and then we quickly finish the bulk part with the part 2, on which I will spend just 2-3 minutes. So I welcome any question now, and we can take a break in the meanwhile.
I wanted to ask you a question. I mean, you talked about it. It's just it went a little bit too fast for me. The way you extract the sample information for sampling. I think it was more at the beginning. I think it's pivot sample that you use.
Yeah.
What is actually happening behind the scenes? I'm not entirely sure I could follow it.
Are you clear with the effect that this has? Basically, since we have a table and is a redundant form of representing the data, pivot sample and pivot transcript will understand which columns are relative to sample and which columns are relative to transcript.
Okay.
And will give us a non-redundant form of information, for example, relative to sample. So
okay.
Here we can see from this pivot sample that we get just 8 rows that are our samples, and all information that we have were existing before or we have calculated that are relative to sample, for example, principal components, covariates, factor of interest, so on and so forth, are outputted. And this is specifically helpful for visualization because we don't want to overplot redundant information using ggplot. So
yeah, definitely. Okay.
Yeah, we want to do it in no redundant form.
Okay, great. Meanwhile, Eric asked, what metric depicts a better picture between transcript abundance and cellular and cellular composition during analysis?
Which metric? Can you repeat the question? Which metric?
Yeah. So what metric depicts a better picture between transcript abundance and cellular composition during analysis?
Okay. So I mean, yes. So one visualization you could do to better understand these two, the transcript abundance, that is the observed data we are doing inference on, and the cell composition that is our infer quantity, could be to plot a scatter plot where marker genes are on the y-axis, for example, CD3G for T cells, and the actual proportions of T cells are on the x-axis. And you could understand, for example, which genes are associated with which cell types.
Okay. Chloe asks how you handle dependencies in cell composition in the PCA. Proportion should sum up to 1, right?
Yes. I mean, there are dependencies. When you start to have from 10, 20 cell types inferred, there are not incredibly strong correlation going on. Nonetheless, 20 is not a big number, so there could be. I guess, I mean, I'm not sure. I have the feeling that PCA can handle it. After all, transcripts are also correlated, so you might have a lot of correlated quantities. But it's an interesting question to think about how PCA handles correlation. But will be the same answer for when you reduce dimension from transcript abundance. Nothing changed from that.
Okay. And I'm not sure if it's a question or remark, but so Claudia responded, proportion are compositional data, not independent observations. No question.
No, I understand. Yeah, of course, all proportions have to sum to 1, so it's a simplex, and necessary all components are correlated. So I understand the problem, and I remark that it's a pretty interesting question. I will research. The question is basically how PCA handles it. You could, first of all, not calculate PCAs on straight proportion, but you can apply logic transformation to transform proportion into unconstrained space. This could help, but still the components will be correlated, but nonetheless, interesting question.
Okay. Niels asked, maybe I missed it, but can one similarity easily get back to a container, such as summarize experiment? Oh, sorry, no, but can one similarly easily get back to a container such as summarize experiment? Sorry for reading.
So summarize experiment can be converted, as I showed before, to a tidy representation. The answer to that question is in the second part of the workshop.
Okay, all right. So we'll get back to that.
There is the actual solution to that. It's quite okay. So I will briefly spend 2 minutes, we don't have much time, on the second part. The second part is about introducing to you a new package that we have built that actually answer this question and make it, these problems goes away automatically in a way. This package is called Tidy Summarize Experiment. And so far I show you Tidy Bulk, which offers a tidy data representation, but especially offers a workflow. Now, what about if you don't care the workflow? You love your own workflow. You just want to harness the power of tidyverse for data manipulation and visualization. The package I will show you now allows you to do exactly this. if you call the tidy function from tidy summarize experiment, you are able to produce a table abstraction representation of your data. This means that you can display your data as if this was a table. You can manipulate it, integrate it, filtering, visualize it, so on and so forth, as we have done before. The difference is that the data is not actually stored in a data frame, but the source of the data is still our summarize experiment object. Okay, so we can query this object as we do with summarize experiment, because it is a summarize experiment. And we can apply all algorithms as we are used to on this object. Okay, so in this case, yes, we have both advantages of keeping the summarize experiment framework and the biconductor framework, but getting all the good thing about tidyverse. I will show you a few things, just a few examples about this. We can filter our data, our summarize experiment object, as if we would do with the table. We can subset it. We can filter it, in this case based on treatment. We can select column, as we have done, we would do with the table. We can produce summary statistics, as before. In this case, we are counting how many transcripts per sample. Again, we can summarize the data, as we would do with the table, using distinct column function from the dplyr. In this case, we are visualizing the annotation of our sample. We can rename columns. As before, we can polish our data set simply applying all the nice tidyverse tools. In this case, we are eliminating the sample prefix. And all this manipulation will be reflected into the underlying summarize experiment object. And in this case, to prove you that this it looks like a table, but actually we can simply reconvert it with the skin summarize experiment. With the skin of summarize experiment, we get back the object. But in this case, we can have it filtered or whatever manipulation we put on it. You can use more complex tidyr functions such as unite. All sorts of complex manipulation done quite easily. Again, some total counts summary. We can combine, of course, pipe different functions. We can group by, mutate by mean count, and then filter by mean count. I mean, this is just example. We can plot the density plot we have done before. This is not filtered data, but you can see here that ggplot works on our tidy summarize experiment as if it were a table. Okay, these are, I hope, answer that question. We are quite proud about this because so far I think the table abstraction is a quite neat idea that we haven't seen before. And of course, this package is about data representation. Tidybulk is about workflow. And as you can imagine, we are working for integrating one with the other. So this, you will be able to use tidy summarize experiment with tidybulk really soon.
Okay, and yeah, there is a remark from Aideen. She says that one of the advantages of S4 classes was to keep annotation and data together, and subsetting options were performed on both simultaneously. Also, S4 objects permit data integration checks. Is it possibly to corrupt the data plus annotation link with tidy?
So with if we are talking about tidy summarize experiment, so the package I'm showing you now, this is an S4 object. Okay. If we corrupt the data, let's say we cherry pick some of these rows, an independent data frame, so you will receive a message saying that this is not anymore a summarized experiment. This won't be a table abstraction anymore. A straight table will be outputted for independent analysis. Sometimes, quote unquote, we might want to subset the data in a way we corrupt it, but we are just interested in some visualizations, so that doesn't matter. Okay, so in a way, if the data is corrupted, S for object is not returned anymore, of course, because we keep consistency. Just a table is returned to us. We can do whatever we want with that, but we will be aware of that. For tidy bulk, where the data at the moment is actually stored in a table, we can do whatever we want, including corrupting the data. But there are 2 points. One is that if we use correctly the functions like left join to join information and proper filtering, we will not corrupt the data. If we happens that we corrupt the data, we will receive, tidy bulk will understand that we don't have anymore a rectangular data set. So we have a kind of sparse data set, and we will receive an error simply. We have a bit of material to go through, but I will show you quite a few things, mainly just to give you the possibility, if you are interested to research further into the matter, as we have a lot of material. Okay, so let's start the single cell part. Not so much, I will not introduce any workflow. The workflow we will use here is from Bioconductor, so whatever you are used to use. Again, we have created a package called Tidy Single Cell Experiment that you can imagine is pretty similar in what it does with Tidy Summarize Experiment. And it allows us, again, keeping intact this underline S for object to do whatever manipulation and visualization you would do with tidyverse. Okay, so we load the libraries. There are quite a few of them. You're free to have a look. For this part, we will use a combination of different peripheral blood mononuclear cell datasets. There are specified here. And this object is a single cell experiment. We have 3,000 cells and roughly 50,000 genes. You can see here, this is a single cell experiment object. And again, we get some information back, but we are quite detached from the data. We don't see so much what's behind unless we start querying this object. If we use the tidy function from tidy single cell experiment, again, we get a table abstraction where the cell-wise information are shown to us. And these are not just displayed to us, but again, this can be interacted with and manipulated with tidyverse. So we get our cell column. Each cell is linked with the file column. We get some properties of the relative to cells, like total counts. And in this case, the cell cycle scores were already estimated. Okay, again, this is a single cell experiment. So if we query it with the function compatible with single cell experiment and whatever analysis we do compatible with single cell experiment, nothing will change. So the functions will never know this is a table abstraction. Again, we are wanting to know how many assays. There are 2, which is counts and log counts. In case we want to add transcript information in our table, in our data set, we can do it with join transcript. This is not anymore a table abstraction, it's just a table. And just to keep consistency, as our friend was asking before, we get a message saying a data frame is returned for independent data analysis. You can see here that this is a table, it's not anymore an abstraction. And we have additional columns as transcript and transcript abundance for each assay. As you can imagine, now we can start to do custom visualization, data integration, and so on. Okay, but let's go on. Again, it's quite easy to polish the data. the data relative to our cells. For example, we have this column with the long file path, and this column includes the data set, the biological group, and the sample ID that we make it joining data set and biological group. And we can use the extract function from tidyr to simply doing that. We are creating sample column, dataset column, and groups column, providing a regular expression with which we extract whatever information we want from the file column. What we get out, again, is our dataset. We can see here we have dataset column, group, and sample column. Again, this is just an example, but you could unite column, separate column, do whatever you wish. Before we go to the next part, which is quality control, this part will use a powerful, probably I reckon the most powerful tool in R so far for functional programming, but not many people know about it. So I will actually launch a poll. How many of you have experience with the function pair nest and map? Nest is from tidyr, map is from pur. Okay, yes, as normally the minority of people use this. I will show you what nest and map do, but some of the code rely also on this abstraction, which is extremely powerful. So I recommend, aside of all this workshop, to really have a look to the function nest and map, as they will make your life so much easier in many circumstances. Okay, to just briefly explain this, we make a simple table. Okay, let's forget about single cell data. This table looks like this. Okay, so let's suppose we want to group the objects that are labeled A and labeled B, and we want to perform some procedure independently for each group. Okay, so one thing we can do, first of all, is to build a nested table. As you can see here, we have now a table within a table. So we have group based on label. You can see here, we have grouped the value column based on label. And now we have this table inside this column, okay? So if we had to extract, for example, the content of data column, we can see here that we get our two the value columns back. Okay, let's see now. We want to summarize this data for each label, and we want to find the mean of the values for each label that are now nested. We can use map to apply whatever function we want iteratively to whatever column we want. In this case, we want to iterate across data column. We want to create a new column called average that is the mean of our value, in this case 3 and 8. Okay, let's suppose we want to filter based on the average. Now we are not just summarizing the data, but filtering based on the max average. And we are unnesting. So before we group the data, now we ungroup the data. And we can see here that we get back our data, but just for the B label that has the biggest average. Another example is we can update the data column. We have grouped the data column. We can update it. So again, we create an average column here. And we can mutate the data column, iterate across the data column, and simply centering the data. So we are subtracting the value, the average to its value. And then we unnest the data, and voila, we have our center value. Okay, this is how it works. So first we map, first we nest, and then we map. This is incredibly powerful. If you start to explore this, we understand how you can do a lot of things without never leaving the data frame framework, the data frame paradigm. Okay, so why I show you this? Because also the table abstraction we are using can be used with nest. And this is particularly helpful when we want to do some procedure that have to be done independently for each sample. For example, inferring the mitochondrial component to filter the possibly dead cells, which are usually characterized with high mitochondrial transcription. Okay, so first of all, we do something we do with our biconductor workflow. Nothing tidy about here. We are just getting our location, which could be mitochondria, NT, from our symbol using some annotation databases. Okay, and we want to apply the per cell QC metrics, which is a utility function from biconductor. And also we will apply a classification whether a cell is an outlier, so has a high mitochondrial fraction. Okay, so these two functions are from biconductor. Instead of separating this data, running a for loop, so on and so forth, we can do everything in a self-contained way. So the magic starts here, where we can treat our abstraction as if it was a table, so we can nest it. In this case, we nest it based on dataset. And so we get the data column that includes our single cell experiment objects. And now we create a new column with mitochondrial information using map function over this data column. And we apply this procedure. Okay, this procedure just calculates the QC metrics, converts the result into a table, and simply annotate this internal table with the high mitochondrial classification. Okay, and does this for each of our samples. Okay, we start doing this. I will show you what we get out of. We have created a new column. This new column includes tables that contain our information we have inferred about mitochondrial transcription. And then what we can do, we can update the data column, joining using left join inside the map as usual, using left join to connect our original data with the mitochondrial information by the cell ID. And then we unnest the data, and what we get out of, sorry, I will show this object here. What we get out of is our data sets, but this, in this case, with additional column with our statistics for mitochondrial, mitochondrial, and so on and so forth, our scores, okay? One thing we can do normally, forgetting about tidyverse when we do quality control, is to plot, for example, density plot or volume plots about our distribution of, for example, mitochondrial information. And there are plenty of ad hoc functions out there that can do this in one line, okay? So you get out a kind of standard plot and you can paste multiple plots together. But what if we want to get out, again, a customized plot, maybe with multiple facets? It's quite easy to build custom plot with this table abstraction. We want to build a faceted plot of three property of our cells, which is the total number of genes for each cell, the total amount of, the transcript output of each cell, the transcriptional output of each cell, and our mitochondrial percent, transcriptional percent. And so we just reshape our data as we would do with any table, and we build in this case, a B-swarm plot with ggplot. And as before, we can customize the aesthetics as we wish, and we build a faceted of these three properties. As we can see here, we have tweaked the transparency, the size, and the color of the points that we want to filter out that have high mitochondrial content. As you can see, usually dead cells are characterized by high mitochondrial proportion as well as low RNA content. Maybe they are leaking, and so they have the majority of the leftover transcripts are mitochondrial. As we would do with the table, we can now filter our data set based on the high mitochondrial label, and this will update internally the single cell experiment object. Okay, we can visualize this object. We can see here that now 100 cells have been, roughly 100 cells have been filtered out. Okay, so we have roughly 20 minutes. I'm not sure I will go through all the material, but I will just show you a few other examples. You are free to pick to get back to this, the website of the workshop to get better understanding of all this. I'm aware it's a bit overwhelming, especially if people are not experienced with this kind of analysis, but still bear with me. Okay, so another important procedure is scaling, as we have done before, and in this case also integration, because different data sets carry a technical variability that we don't want in our data, so we want to account for. Again, usually this procedure involves scaling based on each sample separately, and then integration that will take a list of data sets and we'll put them back together again. I chose to do this again with the nest function. We can see here that we create a new column that is called batch. This new column will be created in the actual single cell experiment object, and then we nest based on this, and then we have a list of data sets. We update our data set with multi-batch norm function. I believe is from either scatter or scran, and then because this fast MNN algorithm does some nonlinear data integration, and this inputs a list and outputs a single cell experiment object. So the input and output are different. This doesn't fit with the kind of tidy framework. And so for applying this, we have from this table, we'll extract our single cell experiment object, apply the function, and tidyfy again. Okay, so until here, we have just used tidyverse paradigm. And just here, we go out a bit, producing a list of data set and then putting that back together. But this is as the actual effect of accounting for this technical variability. Producing adjusted counts as well as producing reduced dimension that take this technical variability into account. This, I believe, will take a few seconds. So if you have any question, is a good time to ask.
Yeah. So do I understand correctly, when doing pooled data, you are actually reformatting the data as it was originally? Like, let's say, you said a list of single cell experiments.
Yes. So pooled data will never be needed if all function will input and output consistent object. Okay. In this case, when you do pooled data, you will obtain not a data frame, you obtain just a list, which is the column. And this will be a list of single cell experiment objects.
Okay.
This function fast MNM once.
Okay. So usually we would say, we would expect like a single cell experiment object, and then we would not need to do that. But here, because this function expects lists of single cell experiments, which is maybe not standard or standardly expected from using the tidy, then you need to go and do this. Okay.
Yes, the problem is that it inputs and outputs two different shapes. It inputs, outputs a unique object. Okay, so you know, for this reason, we have to kind of pull the data out and tidify back. Okay.
Okay.
I would show you how the list look like, but let's see. Okay, because I updated this object. It's fine.
Okay, no problem.
Of a single cell experiment object.
Okay, nice.
And we can visualize this updated object. And we have our cell, our corrected reduced dimensions that we can now use to, for example, cluster, visualize, so on and so forth. Okay, so identifying clusters. In single cell analysis, it's quite important to use procedure that find a cluster of cells in an unsupervised way. And this give us opportunity to find cells that are more similar with each other. And then we can kind of try to interpret and find the identity. So first of all, we apply a procedure that is not anything about tidy. It's just a common biconductor style workflow. It's just, I'm using few pipes here. I'm building a graph of nearest neighbor and building clustering based on this graph. And we are just updating the column labels of this object. Okay, this is not possible to do with tidy as far as I know. So it's just updating in a kind of a standard biconductor way. And we can see here that we have our label added, even though we have used biconductor procedure. Doesn't matter because display, the data that is displayed to us is informatable. So we can see here a further column has been added to our table. And we can do some summary statistics using some tidy dplyr tools, such as count. We can just count the size of the clusters. Another important procedures is again a further dimensionality reduction. Because single cell data is such highly dimensional, there are nonlinear techniques that can applied on top of, for example, PCA, or in this case, corrected reduced dimension, to calculate other reduced dimension that are more able to resolve cell neighborhood. And in this case of UMAP, also able to preserve long distances. UMAP is a function from Skater, so we applied in the same way we would do normally. And the object we are returning with includes our UMAP dimensions. Now, again, plenty of functions out there to build ad hoc scatter plots of the cell into, for example, 2D space. But let's suppose we want to go a bit out of the standard path, build customized plots, or in this case, we want to build a plot visualizing the first 3 dimensions, not the first 2. So we need a kind of interactive way to do this. And because this is a table abstraction, is quite compatible with Plotly, which is an interactive plot framework that is also available for R. And in this case, we are plotting 3-dimension using our cluster labels as colors. Okay, so what we are left with is a quite nice plot that we can explore, we can zoom and so on. One thing I would like to notice is look how much variability the 3rd dimension has. So visualizing also the 3rd dimension helped me a lot in the studies I do and adds a lot of information. So clusters are much better resolved visually and less overlapped if we also visualize the third dimension. And you can better zoom in and realize the relative position of clusters, especially when you have a lot of data. Okay, but this is just an example of having a kind of tidy-friendly data representation can open a lot of possibility with the huge ecosystem that is Tidyverse. Okay, a few last points. After we have reduced dimension and infer cluster, we might want to understand which cell type each cluster, I mean the identity of each cluster, so which cell type each cluster is representing. And there are two ways to do it. One is manual curation of your data, and another one is automatic classification. I will show you them both, and are both complementary. I often do both. So automatic methods usually gets you so far, and then you can really start to explore the data and do some literature review to better understand. In this case, we are first inferring marker genes, which are the genes preferentially transcribed in one cluster and not the other. We use fine markers from CRAN, nothing fancy about this. We are just outputting a list and taking the first 10 markers for each cluster. And so what we are left with is just an array of genes. And then we apply a simply heat map procedure from scatter, so nothing tidy about here. We are just annotating by cluster. And we can see that cells cluster generally together according with the cluster annotation. And we generally go deep into which genes are preferentially transcribed in each cluster and do some evaluation. Okay, this is just an example. It's not that pleasing, but just to show you that normal procedure from bioconductor repository are always possible. Yeah, so feel free to take your break. This workshop is basically done. The thing I was showing you is this automatic cell type classification, but it's just a block of code. We're using Singular. You can have a look by yourself. An example of setting up a pseudo bulk analysis. Don't have really time to talk about that. And that's basically it. Just with pseudo bulk analysis, just for the ones for you who know what is it, it gives the possibility of simulating bulk data from single cell and then using tidy bulk, for example, for further analysis. But anyway, this is for the reading. The workshop is done, actually. Thanks for participating.
Okay, great. Thank you very much.